try to infer the processes acting when a population splits, what happens when a population is bottlenecked or when it expands. So we can use a simple mathematical model to try and describe this process. What I will be focusing on in the first part of the talk is this splitting process here, using experimental evolution, where two populations are exposed to different habitats, and when there is a correlation between an ecological contrast and population structure, we tend to infer that ecology was important during the speciation process. And an experimental approach allows us to actually use a causal approach to this. Usually we do this, as Robert's already mentioned, in a diverse array of organisms. Mostly we've done this in crows over the last decade or so, but we use a series of other bird species, pinnipeds, whales even, where culture really plays an additional role. But recently we've added yeast, where I have been dragged into yeast, I have to say, and I thought that for this audience, yeast might actually be a good or might be a better subject of the talk, or a study organism for the talk than the crows, because many of you actually do deal with experimental evolution as well. So it's a bit scary to talk to you about experimental evolution because I really am a novice, and I've quickly chatted with Christian today that my initial approach was rather naive and we were overwhelmed by the complexities, but well, see for yourself. So the first part will be about experimental evolution in the yeast system. And before I start with the actual bugs, the yeast, let me introduce the main players. That's Sergio, who came into my office about 6 years ago and said that he was working here with Simone Imler on the bottom on a totally different project using fission yeast. They wanted to evolve anisogamy, so it was about the evolution of sex, but that didn't work so well, and Sergio was interested in speciation. He convinced me very quickly that this could be a fantastic organism to study speciation. And then Bart is a great fungus biologist who joined the project and helped us transfer it to Munich as well. And Bernadette joined the project later in Munich and did much of the lab work that you see. The organism is not whale, as depicted here. It's really the yeast. It's fission yeast. Schizosaccharomyces pombe, which is haploid, which is a great advantage for evolutionary studies like this. It's not the baker's yeast that you're familiar with. It has been used a lot for experimental evolution. Fission yeast is the workhorse of cell biologists and has been used to describe cell cycle dynamics in a eukaryotic system. So what I'm talking about you now has just actually accepted for publication and been published yesterday, so it's really fresh from the press. So if you want to learn more about this, just have a look at Nati Colivol from yesterday. All right, here's the background to the project. First, we have to talk about adaptation, because we essentially want to retrace this process. So if there is two habitats, the ancestor has to adapt to the new habitats. And by general theory, which we all know, is there is a variation in the phenotype described here on the x-axis, and there will also be a measure of central tendency. So most individuals here have the phenotype at this value, and if there is selection on this phenotype, so if it has fitness consequences, the distribution of this phenotype will eventually track the underlying fitness function, as depicted here in on top of this. If there is environmental change, which we currently experience with global warming, for example, the fitness function will start to move. And if there is enough variation, genetic variation, then the fitness function will be tracked and result, in this case here, in directional selection or adaptation to this change in environment. Now, we can think about the same phenomenon with a bimodal fitness landscape. So instead of just one fitness distribution or fitness function, we have two that might intersect or not. And if we imagine maybe I can draw that, some population with a phenotypic distribution at a time point 0, then we can well imagine that these individuals down here are dragged into the fitness optimum here in habitat 1, and individuals that lie at the margin at the higher end of this phenotypic distribution attract into the second fitness optimum, such that after a while, again, fitness function and phenotypic distribution match up, and we have, arrived at what we call adaptive divergence. In essence, this is nothing else than just directional selection in opposite directions for two different habitats. Now, where it gets interesting is if we add gene flow to the equation. If there is migration from the red into the blue habitat and vice versa, then we can well imagine that adaptive divergence is impeded. That's the negative sign here, because you end up with, depending on the genetic architecture, of course, with intermediate individuals that fall somewhere in between the fitness peaks. And where this becomes interesting is when we think about how new species can form by adaptation into two different habitats, like the Darwinian viewpoint, really, then we must somehow reduce this negative influence of gene flow on adaptive divergence. So if mechanisms evolve that reduce this minus here, such as associative mating or strong post-zygotic isolation, might be ecological, extrinsic or intrinsic. If that happens and this gene flow reduces, adaptive divergence can again emerge, and then we talk about ecological speciation. So some forms of reproductive isolation have to evolve that reduce gene flow and reduce this negative feedback loop. The thing is more complicated, so here we've just looked at a single dimension of a fitness peak, whereas as soon as we start talking about more traits, we have to consider genetic covariation as well, and evolvability. So in this case, in this sketch here on the left side, I've drawn these two habitats again, two fitness peaks. So you can think of, this is just a hill seen from above, isocline, like in your hiking maps in Austria. And for both traits, so an individual with a high value for trait 1 and a low value for trait 2 would have higher fitness than an individual with a low value for trait 1, for example, and for trait 2. And if this is now the variation that's present in the population, these dots would be in single individuals, we can imagine that it's quite difficult for individuals to reach those peaks because the genetic variation is not present yet in this population. If we look in this scenario here, then it's imaginable that this individual, if it accumulates a few more mutations, can be easily drawn up this fitness peak. The same goes for this individual, so that adaptive divergence can result. Another important consideration is how these two traits depend on one another. So they can be entirely independent. So you might move to the left as this individual, and you might at the same time be able to optimize trait 1. So you can reduce trait 1 and increase trait 2, such that they're entirely independent. But it could also be that it always happens if you move to the left that trait 2 is also, if trait 1 is reduced, that trait 1 is also reduced at the same time. So these are genetic correlations that are described in the gene matrix, and they will determine the outcome of whether a population can move into the fitness peaks or into the fitness optima or cannot. And we can also imagine that the gene matrix actually evolves, that these genetic correlations are broken up during evolution if selection is strong enough. The end result would be something like this, that you have two happy populations sitting in top of their fitness peaks, just as shown previously for the single dimension. Okay. Feel free to interrupt me anytime, right? So it's difficult to get no feedback from the screen, of course. So if you think something is unclear or you get lost, it's better to raise your hand now or just shout at me, probably easiest. And I can stop and we can take a question already at this point. We've now used this basic setup, this conceptual starting point to mimic ecological speciation or to mimic this adaptive divergence under lab conditions. We start with a single population and evolve it in two different habitats, which I depict here always as blue and as red. And the question we ask is, what's the role of gene flow connecting those? So what we change in the experiment is really only the amount of gene flow between those two fitness optima, this multidimensional fitness landscape. We do this with experimental evolution. I don't have to explain that very much to you. You start with something, explain what the something was, and propagate it from generation to generation. And we expect that applying a selection pressure here, exposing it to a new habitat, that the derived populations will be somewhat different from the parental populations at the beginning. The nice thing about pombe, in contrast to Drosophila, for example, is that we can just put every generation in the freezer, a suspended animation, reanimate it whenever we want, and then can test or compete each cycle with each cycle and actually measure the fitness advance between each step. We started from essentially no genetic variation, so that was isogenic strains that differed only in a color marker and the sexual mating locus. And most of the mutations that we're looking at accrued during the course of the experiment, although, as you'll soon see, the decisive experiment that I'm going to mostly talk about started with standing genetic variation that we generated within the framework of the experiment. And this is how it worked. So we have three phases, in phase 1 and phase 2. Well, let's start with phase 1. Phase 1 essentially started with isogenic strains that had no genetic variation and that were only propagated asexually. But they already experienced the two different habitats, a bottom selection and a top selection habitat. Should have been the other way around, I guess, because the bottom is on the top here, but that doesn't matter. So the bottom selection consists in the following procedure. We just grow cells to saturation in a epitope tube, essentially, and then we transfer it to a subset of those cells to a new vial, and just let the cells settle for a certain amount of time. And then we take a part of this bottom fraction that settled down. Right? That's the bottom selection environment. The top selection environment is the opposite, as you can imagine. So we do the same thing. We put cells on top of a vial, centrifuge it, and collect those that remain at the top after a given amount of time. And that looks like a silly, very artificial selection pressure, and it is silly and artificial, but it's quite useful because it's complex. So you can get to the bottom by getting multicellularity, which is what we observe a lot, by changing your cell shape. You can stay at the top by changing your lipid content, by changing your form. So there's potentially many loci and many different phenotypic aspects involved in adapting to those habitats. So we ran phase 1 for about 60 cycles, such that we accumulated mutations, or that mutations got fixed that are relevant for this treatment. And then in the second phase, we added sex to this, because all of this was asexual, and many of them had actually lost or didn't want to have sex anymore, didn't want to recombine. So we then selected for sexual reproduction as well during a second phase, again with many different cycles. And then we pooled all those cells and started phase 3, which is the actual phase that we're talking about now, with standing genetic variation relevant to this experiment. And now the actual experiment, phase 3, modifies the amount of migration or gene flow here. There's actually a difference, so this is migration between the top and the bottom habitat. So the first one is the simplest, of course. We maintain allopathic populations where there's a growth cycle, a growth phase, and ecological selection at the top. Then only top selected cells mate with one another and the cycle is restarted, and the same goes for the bottom populations. The second treatment is a parapatric treatment where we do the same, but instead of just mating top cells with top cells, we transfer a slight fraction, a small fraction of the bottom selected cells to the top and the other way around, such that there is some degree of gene flow here between those two environments. Even more gene flow is given by the local mating treatment, where the bottom cells stay with themselves during mating, the top cells as well, and then they mix. So this is consistent with a habitat imprinted mating, for example. And full gene flow crank up the whole migration river, if you wish, is growth, and then selection, and directly after selection they're combined for mating. That's the sympatric treatment. So here's the experiment in a nutshell. We have 4 migration treatments from low to high gene flow. There is, and that becomes very important, there's 3 life history phases. There's asexual growth, then there's ecological selection, survival in different habitats, and then there's sexual reproduction. We replicated this experiment, of course, with each treatment having at least 11 replicates. And that's, of course, a bit tricky because you have 11 top and 11 bottom here. You have 11 replicates for symmetry. And then we also replicated the starting genetic variation with an alpha ancestry and a beta ancestry. So those are 2 entirely independent genetic ancestries with which we started phase 3. So 2 different runs of phase 1 and phase 2, essentially. We ran the whole thing for this experiment on 53 sexual cycles, plus 13 times as many or so asexual cycles in between. Okay, so what would we predict? And this is where the naive part starts. In theory, you would think that starting by allopatry has the highest chance for adaptive divergence. Right? That's if you have no gene flow, and that's what I introduced. You would expect opposite directional selection, and the populations should both adapt well to their respective habitats, and adaptive divergence should result. Also, this should promote ecological specialization. Each population moves into their own niche. As gene flow is increased, or migration, I have to say, is increased, we would expect less and less adaptive divergence. And since you bring both habitats together at each generation, at some point we would expect the evolution of ecological generalists that survive well in both habitats. That's the theoretical prediction. Okay, let's see what we got. This is now, well, phenotypic is not what you think about a phenotype. It's actually fitness evolution, right? So we measured actual fitness for those 4, or 3, I have to say, life history traits for growth, asexual growth, for sexual reproduction, and then for survival in the top and bottom selection. These numbers here refer to their fitness relative to the ancestral strain, so to the parent. And if it's 1, then the fitness is the same as it was in the starting population, in generation 0. If it's higher, then this is a sign for adaptation. So this is what we got in allopatry. For populations that have been selected in the bottom environment, asexual growth was equal or less to the parents, but populations that were selected for the top clearly had a faster growth rate. The opposite was observed for sexual reproduction, so that the bottom selected populations were good in sexual reproduction and the top selected populations were bad in reproduction. And then as expected, top selected populations were good at top. Bottom selected populations were not much better in bottom. So the selection regime seems to have been stronger here for the top selected populations. Bottom selected populations perform not so well in this top habitat. So clearly, adaptive divergence resulted, and we got habitat specialization. Top being good in top, bottom, well, not so good at bottom. So just one on one side of the environmental niche, if you wish. What's important, though, is that And this is something I think in every system, also natural systems, we should really consider more, is that there's all those selection, all these different life history stages, and there's clearly trade-offs for genetic correlations. So if we look at the correlations here, the correlation is essentially the G matrix here, a visualization of the G matrix, we see that sexual reproduction is strongly negatively correlated with asexual growth. So what does that mean? Essentially, if you take a population here from the top, and that's really good at growth, it will be really bad at sexual reproduction. That's this correlation across the whole all populations. And this is something we know from ecological theory for a long time. You either invest in growth or you invest in reproduction. So you cannot be a superorganism that is good in everything. The rest of those relationships was not strongly pronounced. So clearly the strongest trade-off was between reproduction and growth, but then also between reproduction and top selection. So when you reproduce very well, then you were not so good at top selection in ecological survival in the top environment. Okay, clear so far? Because now it soon becomes quite complicated. Okay, the black box is silent, so I continue. What happened at intermediate migration levels? So now we look at the parapatric and the local mating scenario, and you can already see at first glance there was essentially no adaptive divergence. So we see no difference between top and bottom selection regimes, and if anything, they became bottom specialists because at bottom selection they really increased in fitness and not so much in top selection. There's a slight tendency here in local mating for top selection to be better on in the top environment, but not so much. This may not be so unexpected after this short amount of, just 53 sexual selection cycles and for a mild degree of gene flow. Where it becomes very unexpected was in sympatry. Because in sympatry, we got strong adaptive divergence again. So if we go through this, we see the same pattern for growth as in allopatry. So populations in the top environment, in the top selected part of the sympatric cycle, are doing very well in growth. There's not so much about sexual reproduction, but then what's interesting here is that survival is much better for bottom selected populations, regardless of whether they are tested in top or in the bottom environment. So we essentially evolved an ecological generalist, but there is still divergence in growth as in allopatry. So it's not like the whole population just got swamped by this perfect bottom selected generalist. There is a fraction in this population that is very, very good for top selection, but just during the growth phase. And that's complicated to conceptualize and understand, but it becomes a little bit clearer if you look at the G matrix, which has evolved. It's clearly different in sympatry than it was in allopatry, because here we still observe the trade-off between sexual reproduction and asexual growth, but we also observe a strong negative trade-off between asexual growth and both selection. So you cannot be good in growth and the selection regimes at the same time, and a positive selection between reproduction and the bottom selection. So reproduction and ecological survival is positively correlated. So what this shows is clearly that the G matrix, the genetic correlations have changed under the influence of gene flow, and also that there seems to be a polymorphism, two different fractions in this sympatric setup. We interpret this as two strategies. One strategy is a growth specialist that fares really well during asexual growth. That's kind of this top fraction. And there's another strategy which is really good at survival regardless of which habitat. How they do this and which exact phenotypes behind this, we don't know at this point. All right, this was the phenotype. Again, shout if you cannot follow. For the next step, we wanted to understand what is the underlying genetic basis of this? So do we also observe, or do we observe genetic divergence at the same time as we see this phenotypic divergence? Is that driven by standing genetic variation? Is it driven by large effect mutations or many small effect mutations? And for this, we sequenced all of those populations using a pool-seq approach and just looked at changes in allele frequencies, essentially. If we do this and summarize the genetic variation after selection in the principal component analysis, we see that all 11 bottom populations are nicely separated from all allopatric top populations, with the parent starting strain nicely in the middle. So this really recapitulates this idea of opposite direction of selection. You have variants that just increase in parallel in all bottom populations and an increase in parallel in all top populations. And the principal component analysis, of course, captures most variants that are present in most or all populations, such that this is evidence for ancestral, for standing genetic variation or ancestral variation to play an important role at this adaptive divergence in allopatry. What do we see in the other treatments? Okay. First, some evidence that this might actually be the case is we do principal component analysis of the phenotypic variation for each of those populations and correlate that to the genetic variation. There's a very strong correlation. So standing genetic variation, allele frequency shifts of standing genetic variations nicely align with the change in phenotypic variation. In the other populations, it's very different. So if we look at parapetry, we see those two different pairs, top and bottom selected, and they don't separate. They act as one unit. So gene flow really homogenized them. So there was no adaptive divergence between them, similar to what we've seen on the phenotype. But what is striking, though, is that all those pairs, those 11 pairs of populations, they still encompass the entire range, or even more than that, of these ancestral, of these allopatric populations here shown in the blue and the red underneath. So gene flow between those populations impeded divergence, but it helped to conquer the entire ecological space, or even more than that. So there's even some transgressive element in here. Local mating and sympatric populations, though, in terms of this variation that we see with the principal component analysis, they're all bottom-like. They move into the bottom populations. And that makes sense because that's what we've seen also in the sympatric populations, that they were doing very well on the bottom selection. Okay, standing genetic variation is bottom-like, but how can we then explain the second fraction, the polymorphism, that does good, strategy A, under asexual growth? So there we need new population-specific mutations, and that is what we observe. There's many different mutations, but they're population-specific and hence do not show up on in the principal component analysis. They were not entirely random, so there was parallelism in terms of which genes were hit. So there's a lot of mutations that actually happen in the same genes, but not in the actual same base pair. So what do we conclude from this divergence against gene flow? How can we imagine that this happens in symmetry, that this polymorphism is maintained, at least in generation 53? Well, there's several explanations. One is clearly antagonistic pleiotropy. So what is that? Essentially, if you are an allele of a certain gene in one environment, you do really well. The opposite allele does really poorly. So this would be our top selection, for example, and the bottom selection. Whereas then if you change the environment, change to the bottom environment here, then the allele does good, and the opposite allele does really fare poorly. This is what we observe for many mutations of the standing genetic variation, that they do really good in one environment, but really bad in the other environment. So that the frequency is really high in one environment and really low in the other environment, speaking for very strong selections, and simulations show that this is not, can't be drift, it must be a very strong selection. And then it becomes even a bit more tricky. So if we have several of those variants, we can imagine that they couple their effects and actually might induce reproductive isolation. And this is the model shown here in B. So if you have, for example, one locus 1 that does good, one allele here is just depicted as good in top, and the same allele does bad in the bottom selection regime. We may have a second locus that's closely linked to it, where we get the opposite effect, and they get into linkage disequilibrium, which would, according to Felsenstein, 81, remember this speciation paper, be a very good basis for associative mating, for example, to evolve and reduce gene flow. And we do find several of those combinations where we really have two loci that are in very strong linkage disequilibrium. It's difficult to show because that's pool sec. But one example is shown here on the left in panel A. We observed that, for example, for this REP2 gene, one allele, if that allele is increased as an allopatry bottom, so that seems to be beneficial for bottom, the allele of another gene, the T allele, is always reduced as the parent. So this one is reduced and this one is increased. In the opposite environment, and that's why we're talking about antagonistic pleiotropy, so two loci here coupled. The opposite happens. This allele increases, but then at the same time, this one always decreases for all populations. We can never get into this gray area where both actually would increase. So this is at least indirect evidence that we have strong linkage disequilibrium between the two. This could still be additive, but there might also be epistatic effects, which we have to test then by isolating those haplotypes. So if you think back to this little phenotypic sketch at the beginning, instead of two traits, we have two loci here with the respective alleles, and we can imagine that if we start with a parent somewhere here, you increase the frequency of one locus, one allele for one locus, then you decrease that of the other, and if you get the opposite effect, you may actually reach these two different fitness peaks and reduce gene flow, possibly if you get more of those in the genome. And if you then couple that with associative mating, and I can't go into detail with this, we know nothing about this, so this is just a speculation, this may result in a reduction of actual gene flow in these sympatric populations. What we did observe, and it's a different paper here, if you want to have a look, is that yeast, they have a complicated sexuality, or different from us. So they have something called homothelism, so they can produce hermaphrodites every generation and mate with themselves if they want. And we started the experiment with breaking this complex epigenetic cassette of homothelism into two sexes, a minus and a plus sex, that is stable. But we observed during this experiment, and this is what I mean about naive access to experimental evolution, they re-evolved this cassette and were able to self again. So I think in our populations, we do have a substantial portion of selfing going on, which could contribute actually to coupling environment and with associative mating. Okay, to summarize this part, there was a strong effect of gene flow on adaptive divergence, a U-shaped unexpectedly. We had specialization in allopatry and the evolution of a generalist in high gene flow. But there was a second, apparently a second strategy in addition to this generalist, which was doing very well, was a specialist in growth. These trajectories were strongly dependent on the existing trade-offs or genetic correlations. We've talked about this, and we've also seen that those genetic correlations responded to selection exposed in those different environments and changed during the course of the evolution experiment. And candidate processes, and this is still a big question mark, might be antagonistic pleiotropy in several loci, and associative mating that might explain why we observe this polymorphism still in symmetry. Okay. Okay, so this is now another view in hybridization, this time in natural populations of fission yeast, not experimental evolution anymore. And there I quickly introduced the idea or the theory again. So if we have migration as we just induced experimentally, there's different phases to that. The migrant may just be selected against or die, or may be able to mate with a local, leading to hybridization. And then if this hybrid survives and back crosses into the original population, we get introgression. And then if the migration is successful, we talk about gene flow, right? And this hybridization can have different outcomes. There might be speciation result, that there's actually some barriers evolved to this gene flow, as shown here on the left side, and reinforcement may further this process. There may be speciation reversal, so the gene flow is so big that you go back to one single population. Hybrid speciation might result like a third independent lineage from a certain combination of those two genomes may lead to a lineage that's reproductively isolated from both of the parents or the evolved parents, or you might get an intermediate stage of diversification in a stable hybrid zone, for example. And key to all of this is, of course, reproductive isolation evolving in one form or the other in pre-zygotic or post-zygotic status. And usually how we study this and how we did that in crows and whales and all kinds of things is that you just scan the genome and you look for regions that are more differentiated than others, assuming that the other regions have been homogenized by gene flow. And so if we think about population evolution over time, mutations might accumulate or might pop up that are good in one population rather than in the other, so that there is divergent selection, as we've just seen in the experiment. The rest is still broken up by recombination. There's a chance for gene flow. And then over the course of time, you might develop larger areas, new mutations accumulated in those regions that might show linkage disequilibrium. And then overall, gene flow between those two populations also ceases or decreases to a point where we have full reproductive isolation. So that's the typical approach from in natural populations. You have predefined entities, you have population 1 and population 2, you compare them and look at the genetic differentiations, divergence across the genome, and then try to infer, is genome, in which kind of stage of this process are they? And this was attempted in a global collection of Schizosaccharomyces pombe strains from Dangifaris here in 2015, which is a fantastic sampling scheme across the entire globe, here shown by different colors. And what's interesting about pombe is it's always human associated, so nobody knows where it really comes from, where it's derived from in the wild. So we find it associated with wine or palm or fruits, grains, molasses and sugar, often associated with, agriculture, essentially, human agriculture. It's also used for beer brewing in some regions, but it doesn't taste so nice as Bart can tell you. He brews every Christmas, he brews pombe beer for us, and everybody prefers the normal baker's yeast beer in the end. So they then did a population genetic analysis on this with 161, I think, strains, many SNPs, and it turns out that there is absolutely no structure. There's no structure by geography or by ecology, by where they grow. And that is puzzling, right? I mean, they live across continents. They have very clear adaptation to certain habitats, and there was no structure. What makes this even more puzzling is that they show very strong reproductive isolation. So if you just plot the number of the divergence between two strains and look at the proportion of viable offspring, you clearly observe the closer they are, so the more similar they are, the more viable offspring they have, and then you get really strong post-zygotic isolation at some point. But that contrasts with this picture of a totally panmictic, happy yeast population worldwide. So when I saw this, I thought, well, couldn't it be possible that we actually look at a hybrid swarm, that there is very divergent genetic material in there that actually has strong reproductive consequences, but they're just jumbled up by admixture. And this exactly turned out to be the case. So we started doing population genetic analysis, not genome-wide, but chopping it up into windows of 200 SNPs. And that's what we observed in a principal component analysis for all those strains. We've always seen two very, very clear clusters. One is the blue cluster and the red, so it's nothing to do with the experimental evolution, right? It's also just blue and red. So we have a blue cluster and a red cluster. And the blue cluster always showed much more variation on the second PCA, principal component axis, as well. So we have a high diversity blue cluster and a low diversity red cluster. And that explained these two groups explain 60% of the entire genetic variation in this window, which is really high. And then we went on window by window, and for each window we defined, so this population, for example, in the next window might jump to the red cluster and the other way around. And if we do this across the genome, we get this picture. So we get strains like this GB869 here at the bottom that are always in the red cluster and the low diversity cluster. And then we get others that are almost always in the blue cluster and the high diversity cluster. But for most of them, it's a mess, right? They're entirely admixed between those two clusters. And the proportion of variation explains, I didn't lie to you with this PCA here, it stays constantly high for the first and the second principal component. So the entire genome really falls into those two blue and red clades. And if we then use simple hierarchical cluster analysis, we can actually make sense of those. So we see patterns emerge, similar strains, and define those clades, which weren't really visible before. Even with fine structure, you couldn't see them in the same way. And what you also observe is that the chromosomes behave differently. So one column here is a chromosome. They mostly tell the same story, but not always. Looking further into now the divergence per window between red and blue, we observe that the divergence measured here as dxy is 3 or 15 times higher, depending on the blue or the red clade, than the diversity within the blue or the red. And that clearly suggested an evolutionary scenario like this, that we really have, we had speciation of the blue and the red before. They diverged for a long time, probably without gene flow. And at some point, we had a pulse of hybridization that then led to this genome-wide admixture. And we ran simulations on this and can show that a model with constant gene flow over time does not work at all. So we need allopatric divergence and then a pulse of hybridization. We also used models to estimate when this hybridization happens.
Excuse me, the pure blue and red ones, are they geographically clustered or not?
A little bit clustered. So the one is more associated with Europe and one is more associated with Asia, the actual pure ones where you find them. Whereas in the Americas, they're totally admixed. So in the United States, or like in the whole Americas, you only find admixed strains. And that's actually why we think, and this kind of is consistent with our estimation. So we estimated that 20 to 60 outcrossing sexual generations, this hybridization event happened. Very difficult to time in years because we don't really know how many sexual generations a wild yeast has relative to asexual generations, but just digging through literature, this is rather consistent with hybridization in the 15th century or something, where humans started transcontinental trade. So we think that one strain was brought into Europe through Marco Polo, essentially, right? 12th, 13th century, admixture happened, and these admixed strains were then transported into the United States, or the Americas, I have to say, not just the United States. This is, of course, high speculation, but it's at least consistent with this data. Divergence somewhere in natural populations, and then human-associated trade. And what's interesting and of more relevance than just the history of the species is if you look at the heritability of 228 phenotypic traits that have been measured, and that's biochemical traits, actual cellular phenotypic traits, and many others. We observed that the heritability estimated using all SNPs, and that's 100,000, versus our just 2,000 haplotype blocks across the genome is exactly the same. So we capture all the phenotypic variations with just considering the ancestry proportions of a strain, which means that after hybridization, the mutations that happened after hybridization contributed hardly anything to phenotypic variation. And that's also seen here in this PCA, where the almost pure blue and the almost pure red are at least slightly divergent, but all the hybrids that encompass, or the admixed strains encompass the entire diversity of both parents, and even more so, suggesting transgressive phenotypes. And if you look at those phenotypes in detail, we can, at least with evidence for dominance in the red clade for some traits, dominant phenotypes that are where the blue background is dominant. Some are intermediate, so the admixed strains are intermediate between the pure ones, and then some evidence for admixture actually causing transgressive phenotypes that are beyond the variation of any of the parents. The same goes for reproductive isolation. So whether you use hundreds and thousands of SNPs or whether you just use ancestry proportions, we observe exactly the same picture. So the more similar two strains are in terms of ancestry proportions, so two very blue ones, they will have a high spore survival. But the more red the other one gets relative to the blue one, the less spore survival you get. So you clearly get reproductive isolation just based on those ancestry components. The next question we asked, and it's not going to be very long anymore, so I hope you still haven't didn't slumber away. If you look at the ancestry proportions across the genome, they're very, very uneven. So most of there's more blue in our sample, but there's some parts of the genome that are almost fully blue and others that are almost fully red. And this doesn't look like just, it could be random, right? But it could also be that there is strong selection, that there has been a mutation here that has been really very beneficial for if it's blue, if it's a blue background, and here it's very, very beneficial if it has a red background. The primary candidates for this are structural variants, because they often have very strong phenotypic effects. So we categorized structural variants for many of those strains and all those different clades that we found by hierarchical clustering. And a quick take-home message is never do this with Illumina. It has been done before by Dan Jafaris, and using long-read, both Oxford Nanopore and PacBio, de novo assemblies or mapping approaches, there is hardly any overlap with the Illumina called, really well done Illumina called structural variants. So I don't believe any structural variants anymore that are not based on long reads. Because yeast is a small, tiny thing with not much repetitive elements, right? And what did we see? So is it really that structural variants, indels or inversions, swept for the blue in this part of the genome, for example, and for the red in this part of the genome? Doesn't absolutely not seem to be the case. If we do look at the 2D side frequency spectrum, so where we look at the allele frequency in the red clade versus the allele frequency in the blue clade here, we do this for SNPs. We do find quite a few high frequency derived for the one or the other clades, so in red or blue, which would be evidence for hitchhiking or positive selection. And we find nothing of this in the structural variants. So even in those regions that are all blue, you don't have a high frequency of any of the structural variants. On the contrary, this comparing those side frequency spectra suggests that there's strong purifying selection on all structural variants. So why do we observe this pattern then? The second idea would be that there is ancestry disequilibrium induced by epistasis. So if you're blue here, you also have to be blue here, because otherwise there is an incompatibility and negative epistasis. And there's some evidence for this, because if we compare all blue-blue comparisons to all red-red comparisons and to all mixed comparisons, we clearly see that the linkage disequilibrium is much lower between the mixed comparisons, meaning that there is more blue-blue and more red-red combinations than expected by chance. So there is possibly some role of epistasis, some kind of Dobzhansky-Muller-like incompatibility that caused this landscape. Okay, I'm through. Conclusion here. All known strains of S. pombe, with the exception of, actually, luckily, the original Leopold strain that was selected for all the cell type work, it seems very pure. So most of them are recombinant hybrids from at least 2 ancestral lineages. The ancestry components explain all the phenotypic and reproductive variations. We don't need to look at individual SNPs. A block here is enough. There's no evidence for structural variations influencing these genome-wide patterns of that mixture, but there might be some role for epistatic selection. That's it. So thank you very much. Beautiful picture by Sergio.